Bonjour tout le monde. Bon matin. Good morning. So we are going to start our plenary session. On va commencer la session plénière de sciences fondamentales. Bienvenue à tous. Donc on va commencer et je laisse la parole à ma collègue Petronella pour présenter notre invité.
Okay, good morning, everyone. I would like to thank you being present here, and I hope other people will come soon. So my job is to introduce our keynote speaker today for basic science, Dr. Susan Moir. Dr. Moir is currently a senior investigator at the Laboratory of Immunoregulation at the NIH in Bethesda, USA. A little bit about her curriculum. In 1996, Susan Moir received a PhD in immunology and microbiology from the Université Laval here in Quebec City. The same year, Dr. Moir joined the Laboratory of Immunoregulation at the NIH as a postdoctoral fellow under the mentorship of Dr. Anthony Fauci. In 2015, she received a tenure-track position, and in 2020, Dr. Moir became a tenure senior investigator and chief of the B-cell immunology section at the Immune Regulatory Laboratory. During her career, Dr. Moir published more than 100 milestone publications, research articles, and reviews listed on PubMed, and contributed to multiple books in the field. She received various awards in recognition of her professional performance. I can list here in 2014, she was the Trans NIH Early Stedman Tenure Track finalist in immunology. And in 2020, she received the prestigious NIH Director Challenge Innovation Award. Dr. Moir has a unique internationally recognized expertise in B-cell biology, especially during HIV infection. Dr. Moir, we thank you for accepting our invitation to speak today. The title of your presentation is The Role of B Cells in HIV and SARS-CoV-2 infections. We are keen to learn about your most recent discoveries relative to humoral immune responses against these two challenging yet mechanistically distinct viral infections. So, and before we start, we have a small present for you, so I hope you'll take it. So this is for you. Thank you. Thank you.
All right, so I'm going to speak off of, hopefully that'll work well. So thank you so much, Petronella, for the kind introduction, and for the organizers, the kind invitation. I'm honored to be here today, and this is my hometown, so it's a pleasure to welcome everyone to Quebec City. I hope you get out there and enjoy the sights of the city. Je voudrais remercier les organisateurs. Je viens pas souvent à Québec, mais j'essaie de garder mon français, alors j'espère que la présentation va vous plaire. Les diapositives ne sont pas très belles, certaines d'entre elles, mais je vais essayer de vous naviguer le tout. Alors, okay, back in English now. I don't have any conflicts to disclose. These are my learning objectives. I'm going to give a little background on B-cell immunology, and then some differences between that we've described in HIV infection, and then some of the differences between SARS-CoV-2 and HIV, and also describe some the different populations that respond to both SARS-CoV-2 and HIV. So I've divided the talk into two sections. One will be on studies in chronic HIV infection, and the second part will be on our recent efforts with regards to SARS-CoV-2, mostly in response to vaccines. So just to walk you through very quickly on the immunology of B cells. So I'm going to try. Actually, let's see. No, I'm just going to talk, and then hopefully I'll walk you through. So as a naive B cell encounters its pathogen, it has a couple of different choices. On an early response, it'll usually go through these short-lived plasmablasts that do contribute to early serum antibodies. And then the B cells also can enter into a germinal center reaction. This is the preferred route where they generate long-lived memory B cells and also long-lived plasma cells that then are home back to the bone marrow and are the sources of hopefully long-lived as well serum antibodies. So in the context of HIV, there's a lot of perturbations that occur, and I've shown they're described here as B cells develop out of the bone marrow. And I'm not going to talk about most of these that we've described and others have described over the years, but I will focus on two areas. One are studies on lymph nodes in HIV infection, and the second one, and I'll spend a bit more time because that relates to SARS-CoV-2, and that is looking at the different memory B cell compartments that arise and those that are perturbed in HIV infection. We use a lot of flow cytometry, and I just want to draw your attention. So a lot of B cells in the context of HIV infection downregulate a complement receptor 2 or CD21. And years ago, these are very easy to see when they're amongst short-lived plasmablasts. But when they are not, it's a bit more difficult to know what's going on. So and sorry, I apologize, these are some of the problems that in a transfer from Mac to PC. But 2005, Max Cooper, a stellar immunologist who discovered B cells, described this paper that was published in 2005, where they described these altered memory B cells or unusual memory B cells that were defined by the expression of an immunoreceptor called FcRH4 that was later renamed FcRL4, and it's an immunoregulatory receptor that's thought to have inhibitory functions. But two of the striking features is that these cells had low expression of the complement receptor CD21, as well as high expression of CD20. And so when we saw that, we thought, oh, we have some of those same cells in peripheral blood of HIV-infected individuals. And so that's when we published back-to-back. They were more looking at gene regulation and gene transcript profiling, while we were looking at similar B cells in HIV infection, and we saw them in the peripheral blood. We named them tissue-like memory B cells because in recognition of the work that Max Cooper have done, and these are circulating B cells. So if you look at HIV viremic individuals, these are the cells here that we describe as low CD21 and low expression of the pan B cell marker for memory B cells, which is CD27. So the profile is CD19 high, CD20 high. Those are two markers that define B cells. Expression of FcRL4, expression of an integrin receptor called CD11C that's usually seen on dendritic cells and not B cells, and the expression of the inflammatory homing receptor CXCR3. So these cells here are very minimally expressed in healthy donors. However, they're not absent. We also demonstrated several years later, as many other groups have shown, that these cells also express the transcription factor for Th1 with TBET, and the highest expression is seen among TLM cells. So over the years, many groups have described in different contexts of exposure, whether acute or chronic, where these B cells arise. And so this is a nice review that was published just not long ago, describing, sorry, I can't see very well, but so describing that in the context of a Th1 type, so interferon gamma, you induce the expression of TBET that then induces the expression of all these other receptors, and that these cells come up during the acute phase, but then subside if that acute phase subsides, but that they remain high if they do not. And that's where you get these chronic and almost, in the case of HIV, somewhat exhausted B cells. And this is not only seen in HIV infection, but also in other chronic conditions of immune activation, such as autoimmune diseases, some parasitic diseases, and even in TB. It's interesting, too, though, that these cells do have a normal component. And this is a very nice paper that was published in Science, basically describing the atlas of tissue human cells. And this was done on organ donors who were deceased, but they were otherwise healthy, as much as we could tell from the paper. They used single-cell analyses to describe many many phenotypes of immune cells. And interestingly, one of the ones that popped up are these tissue-like memory, which others have called different ways. These are age-associated B cells. That's mostly seen in mouse models. But they have the same features that we described and others have described. And in healthy individuals, they are found in the liver, bone marrow, and spleen. So they probably do have a normal, healthy function that remains to be described. So several years ago, we were interested in understanding where are those cells residing in lymphoid tissues, and we were able to complete a study looking at inguinal or axillary lymph node biopsies from HIV viremic and HIV uninfected participants. We did several different things, extensive phenotyping and looking at antigen specificity using tetramers. We did some transcriptional profiling. We did some confocal microscopy with histocytometry to quantify some of the location of the cells we were interested in. And we did some material analysis looking at B cell receptor repertoires. So I'll walk you through a few of those. So this is a very busy slide and doesn't quite convey as well as I would like it. But the most important, so we did this was bulk sequencing on sorted cells. I just want to draw your attention to the profile that we were able to demonstrate for the HIV-infected, HIV-specific memory B cells. And they had some of the very same features that we had described in the peripheral blood. And basically, that's that they express high levels of CD19, high expression of FcRl4 and its related immunoregulatory receptor FcRl5, as well as CD11C, low expression of CD21, high expression of CXCR3, and of course the telltale sign, the strong expression of the transcriptional, of TBET, sorry. We also wanted to look at their location in these lymph node tissue sections on the left. I think your left, yeah, is a healthy individual. So you can see that the expression of TBET is located, but in the subcapsular area, and so this is. We think that they probably migrate in and are close to the sinus macrophages and play a role in sentinel or uptake of antigen. In HIV-infected individuals, especially those who are viremic, you see the lymphadenopathy, where these giant germinal center germinal centers occur, and these are shown here. They're reactive by the expression of Ki67. And so when we looked at the expression of TBET, it was interesting that there was a more diffuse expression, but it was really excluded from germinal centers. And so that's shown here, where we found, using the histocytometry capabilities, we're able to define the different zones and demonstrate that most of these TBET-expressing B cells were outside the germinal center. And we were also able to demonstrate that the higher the expression of these cells, the higher frequency of these cells, inversely correlated with the capacity to neutralize HIV. So basically, the current model for looking at TBET in the context of chronic immune activation is that those B cells that are activated by the cognate T cells, where the skewing is a little too strong of the Th1, either never make it into the germinal center, or if they do, they don't spend a whole lot of time in the germinal centers where they acquire affinity maturation. So this is where, we've positioned these cells basically not contributing very well to the neutralizing capacity to neutralize HIV. So as summary for this part, CD19 high, TBET high memory B cells accumulate outside germinal centers in chronic HIV viremia, and they are similar to those that we described in circulation, so in the peripheral blood, and they account for the majority of HIV-specific lymph node memory B cells. Their non-GC location suggests that they receive diminished T cell help, and as a consequence of that, they have reduced affinity maturation, and that has been observed in many different studies where few people make neutralizing antibodies against HIV. All right, so I'll switch gears and spend the rest of my time talking about B cells and SARS-CoV-2, but I'll circle back to HIV a few times. So as we were going into lockdown, as most of you, we were not able to do our HIV work. And so as it took us a little while to reorganize and then start to look, do some studies on SARS-CoV-2. In my group, we were able to, we decided that we were going to start looking at the response of B cells to the 2-dose SARS-CoV-2 mRNA vaccine. The people who were enrolled were mostly NIH clinic and lab employees who were allowed to be on site, eligible to receive their 2 doses. And it just happened to be that the one provided at NIH at the clinical center was the Moderna vaccine. So we had the 21 individuals, and we followed them longitudinally. The demographics are shown here. The most important is that through the 6 months of analysis, 20 of them remain uninfected. We did serial blood draws at the baseline of dose 1, and again after, and at the baseline of dose 2, and several time points in between, because we wanted to look in real time at what was going on with the B cell populations in the peripheral blood, and a lot changes. And then we came back, and they all came back at month 6. So just to, when we want to look at responsive B cells and looking at spike-specific B cells in response to the vaccine, we used the spike, it's called S2P, it's the one that was the mRNA encoded trimer, and we created tetramers, either with the full S2P or the S1 within the subdomain S1, or RBD within S1. And with those tetramers, we incubated them with B cells along with a bunch of other antibodies that recognize B cell markers. And we looked at the frequency of this one showing here of plasmablasts, and they come up a little bit after dose 1, and then there's this rapid spike, very strong response after dose 2, and it occurs very early on. And that's why if you look just a little too late, you'll miss that very strong response, because it comes right back down very quickly. And the second phase is looking at the B cell response, so the non-plasmablasts, and those, so technically these are the memory B cells. And again, you see a little smattering after dose 1, and a pretty strong one after dose 2, but it also comes down, it also wanes. But interestingly, I'll come back to that in a little bit. So one of the things now, we use spectral flow cytometry, which is a really good way of being able to expand the number of markers that you can look at. So in this case, we actually only had 23 markers, which is actually on the low end. But we were able to do some this clustering that's now very common to do when you have a lot of markers. And we identified 30 different clusters, among which some of them were the ones that were expressed, that were binding the spike proteins, or in this case, RBD and S1. And we focused on those clusters to then identify which ones were the most persistent. So to be able to do that, I want to as many have reported, that there's actually a waning of antibodies after each dose, and especially over time. And if we take the last 2 time points, so at day 60, the day 60 from the first dose, and then at month 6, when you look at the serum antibodies, there's a significant decrease over that time period. However, when you look at the memory B cell frequencies over that same time period, it actually increases between month 2 and month 6. And that is the intersection of B cells that have been highly activated, that then disappear, but then you establish a nice resting memory population. And that's what this conveys here. We're not the only ones to describe that. John Ware's group also, and a few other groups have described that. So it's not, it's been commonly reported. But one of the more interesting things is that in this, among the cells that we found the clusters that we found to be responsible for this stabilization of the response and the longevity of the response is this cluster 2 that has a resting memory phenotype that doesn't express CD11C, that's a surrogate for TBET. And these are the cells that are unfortunately depleted or decreased in HIV viremia. So this is a graphic, a little bit of the graphical artistry of our findings, because we also wanted to establish correlations between the cellular response, B cell response, plasmablasts and memory B cells, and endpoint antibody titers. And so through a lot of modeling with the help of our bioinformatics team, we're able to identify that many of the plasmablasts, or the major plasmablast populations, were correlated with especially shown here, there's a very strong correlation both at day 56 or after vaccination and even up to month 6, that early response predicted that antibody response way out to month 6. And so that's a nice correlate. The unfortunate part about this is done on fresh cells. And when you do it from frozen cells, plasmablasts just don't, they are very fragile and do not sustain very well after freeze thaw. But the memory B cell population, though, was also correlated, and that's shown here with different populations. The one that didn't correlate is the one that is highly activated and does come up. You see it come up strongly here in blue, but it doesn't correlate with the antibody response. We think it really just reflects a very strong immune activation, but it's also very variable and probably has a lot of underlying factors that just don't then correlate with the antibody response. To gain a little bit more insight into the different populations that get activated, we have done some single cell analyses. And so this is, I have to say, was a hard slog, but nonetheless, because we just don't get enough cells, and we wanted to look at the different populations at the single cell level, and we were able to find 3 individuals of our cohort on whom we had enough cells. These were all blood draws, so that we don't get that many cells. And so we isolated plasmablasts, we isolated the antigen negative B cells, and then we isolated the antigen positive B cells, those are S2P positive. And from those, we did single cell processing, and we did the 10x and looked at gene and protein expression and B cell repertoire. I'm just going to talk a little bit about, so if we take all time points and all cells, we had about 110,000 cells to cluster, and this is clustering at the transcript level. And so what was obvious pretty quickly, we identified 7 different clusters, but one of them really stood out, this one here, that was not present in the other populations. And so we did, and that is also the cluster that we found to be particularly expanded on the right side here. So this population here is cluster 5 that came up strongly and so was very responsive to the vaccine. So we decided to recluster that one, and that's shown here. And so from cluster 5, we generated another 5 clusters, and those were enriched with those that were antigen specific. And from that, we were able to, we just looked at where in red are shown where the spike specific response is occurring over time. So you see at early time points a little smattering in this area, and then it starts, and then by day 28, you start to see the segregation that occurs. And so, and then by month 5 after vaccination, you really see this clustering that really is up in this corner here. And that is the clustering of cells that have longevity, and they're the resting memory B cells. And so we also did trajectory analysis, and what we identified was a population of activated B cells that really contribute to the resting memory B cells at month 6, and they are not in the population of those TBET expressing cells that we and others have described. This is just to show, again, at month 6, when we're looking at those populations that contributed And this is called some switch analysis, and I'm not going to go into any details because it's done by the people who do repertoire analysis. And so they looked at how often does one population clonally relate to another. And what we found is that there was this population here of these CD71 activated populations were the one that most contributed to the longevity of the response at the end of the analyses. So we do need to revise this model because it's getting to be more complicated than just a couple of simple markers, and that's what we hope to be working on over the next little while. And so again, I think this model is a bit simplistic. It's very helpful, but it needs more work. So I'm just going to spend the rest of my time talking about our most recent data. And that is when we looked at the effect of the interval between infection and booster vaccination in individuals who were in a different cohort, but some of the people were also enrolled in the first cohort. So in this cohort, we had 3 groups, and we had one group that remained uninfected throughout the analysis, one group that was infected prior to receiving their boost, and then one group that got infected after receiving their boost, so a breakthrough infection there. And we did a bunch of different analyses. Just to go through the demographics, in total we had 66 participants, 44 remained uninfected, 11 had a prior infection to getting their booster, 11 had the breakthrough infection, and this was during the first Omicron wave, and so it was totally unexpected that we would gather the same number of people in both groups. And again, we did serial blood draws, but I'm just going to talk to about 3 time points at the baseline of receiving their booster vaccine, and then 30 days later, and then 60 days later. And again, I'm sorry, this is where the color coordination breaks down, but in the white color, the loss of color, is the uninfected group. And basically, there was no difference in the demographics at the gender level, neither which booster vaccine they received, Pfizer or Moderna. We did find a slightly lower age in people who got a breakthrough infection, so they're post-boost vaccine, but otherwise there were no other differences in terms of the time between the dose 1 and their booster. Again, we were looking at different metrics of B-cell responses. One looking at the serum spike binding, and we did some pseudovirus neutralizing antibody analysis. We also took the PBMCs and we put them in culture to get them to polyclonally stimulate them, and so that's shown here. And from those cells, those memory B cells become antibody secreting cells, and those antibodies can then be measured by the same way we do it in the serum. And the other one is to look at spike-specific B cell by flow cytometry. I'm just going to walk you through. Again, there's a problem with the color coordination, so please forgive, but I think you'll get the gist of it. This was supposed to be purple up here, and it doesn't show up. But basically, when we were looking, I'm just showing some, we looked at a bunch of different response to variants, as well as the first one, which is the ancestral. And you can see a pretty strong induction between day 0 and day 30 that then stabilizes. And if we just go and look, though, by group, then that's where you can start to see the differences. So whereas the uninfected and those who had a post-boost infection had a very strong response, we started to notice that those who came in with a prior infection really didn't respond very strongly to their vaccine. And that's shown both at the binding antibody level, but also serum neutralizing antibody levels. The difference was even more striking in that there was absolutely no increase over time for people who were prior infected. And when we looked at the culture supernatants of the memory B cells that we put in culture, we saw a very same trend as with the neutralizing antibodies. And as you can see here, there's increase between day 0 and day 60, no increase for the prior infected, and an increase of people who were, had a breakthrough infection after their heart. So the nice thing about doing B-cell analysis by flow cytometry is that you can look at a variety of different responses in combination, which you can't easily do when you're just looking at antibodies or even secreted antibodies from memory B cells. So I'll just walk you through. B cells are identified by the expression of CD19, and we do this little thing to exclude all cells, all naive cells, because they do contribute some background binding. And from that, we use our tetramers that we then incubate with the B cells. And I think I can do this. All right. So we were looking, to increase our capacity to look at a specific response, we tend to look at the response of S1 and RBD. And this diagonal here is those cells that are binding both of them. And because RBD is within S1, this is our specific. We know that this is a nice specific response. In the top panel here, you can see on the right, these are the cells that are binding to both the ancestral and the, in this case, we were looking at BA1, so the first Omicron variant. And those are the ones, so this is where you would see an increase in breadth, and this B cells that bind both. When we looked at our different groups, we saw the same thing as we saw with the antibodies, and that is a nice increase of people who were uninfected or post-infected, and basically a very flat line of people who were prior infected. What I should say, though, and I didn't explain that very, very well, is that those who are post-infected tend to come in with a high response, and it's not that they have so this concept of hybrid immunity, I don't dispute it at all, but they come in with a high response and they just don't have a further response. And that is broken down into two groups. And so while people who were uninfected and post-infected, their response to the booster was strong, and those who were prior infected, what we saw we could see two types of responses. One that almost looked like the other two groups, and that is they did have an increase between day 0 and day 60, but the other basically had the flat line. And where we saw a really poor response to booster is people who had been infected within 6 months of having received their booster vaccine. And so what we ended up doing, this cohort was small. By the time we whittle it down to breaking down each of the groups, we focused in on 6 individuals in the prior infected group who were less than 180 days from between infection and boost. And we matched them with people from the 6 from the uninfected group who had exactly the same demographics at baseline and response to B cells. So we really tried to match them because we knew our group, our cohort, by then was very small. And just go back, sorry. Oh, no, I won't go back. All right, so what we were interested in trying to find out is whether this lack of response or this poor response is intrinsic to B cells. And we had an inkling of that from the culture data. And one of the ways of looking at B cell responses is to look at incubate B cells with something that's going to trigger their BCR to go and look at the signaling events that go downstream from the B cell stimulation, BCR stimulation. So 2 of the downstreams that we look at that are often looked at for B cells are tyrosine kinase SYK and PLC gamma 2. And so we looked at their phosphorylation levels, and this is nice, can be done in flow because it takes about 2 minutes to do. So your B cells remain intact, and the other nice thing that we were able to do is that while we stimulate polyclonally through the B cell receptor, you can look at the response, basically the antigen-specific response. And just to walk you through a little bit, if you take untreated, you see no phosphorylation. So we're showing phosphorylation of SYK and phosphorylation of PLC gamma. And you want to look at the induction after triggering with anti-BCR. And this is showing someone who was recently infected at baseline, a very strong response of the antigen when we gate on the antigen-specific B cells, but also on the non-specific B cells. And by day 60, that response had gone down by half. So they were very active and poised to respond at baseline. And that's reflecting that they were just recently infected. So their cells are still kind of revved up. But once you try to stimulate them again by the vaccine, they don't have such a strong response. So that's shown here in the group of 6. So you can see that the biggest difference between the uninfected group and the prior infected group basically is at baseline, where in the prior infected group, their capacity to respond to BCR stimulation is high, but actually goes down over time, whereas in the uninfected group, their capacity to respond to BCR stimulation is stable, and that reflects the nature of their B cells. And so that's shown here. One of the best ways to really report this is to do the ratio of the response between day 60 and baseline. And this one goes down in the prior infected group and stays very close to 1 in the uninfected group. And the strongest response we saw was from PSIC. And we actually saw a very, very, very strong correlation between this ratio and the interval between infection and vaccination. So that means the shorter the interval, the poorer the response to the vaccine. So finally, we wanted to drill down into whether we could identify a population of B cells that could be responsible for this difference of the BCR signaling, and that's shown here. So if we go back to the model where we do see an induction of CD21 low B cells right after being stimulated, in the days after being stimulated. So basically, we're looking at, in this phase here, you would expect, okay, we probably will see a difference in CD21 low B cells. That is not what we found. So in the uninfected versus prior infected, if you look at baseline, so these are CD21 low, there was absolutely no difference. When we looked at day 60, we did see a difference, but it was not, it was in the CD21 low, and the difference, though, is in another population. And let me try to explain that a little bit better. So if we start with CD21 low B cells at baseline, in the uninfected group, that percentage goes right up between the baseline and day 60. And that reflects the fact that they get activated. In the prior infected group, they do go up, but very marginally. And the difference is at the last time point, not at the first time point where we saw the difference in functional response. But when we look at this other population, so these are the CD21 positive, CD27 low, and these are the cells that are in this quadrant down here. And we think that when B cells go into kind of, they're resting, a lot of these, especially after the first 2 doses, they downregulate their CD27. That makes them more resting, but better poised to respond to the booster. And so to nail that one down a little bit, we took the B cells from in the 2, we just broke them down into the 2 27 positive and 27 negative, and it is the 27 positive. So the more you have these cells, which are the ones that are found to be the majority of the cells in the prior infected group, the higher that response. So if you think about it, they were responding highly at baseline, but by the end of the time, at day 60, that number, those 2 populations had normalized. So we think that that's what at least partly explains the differences between the 2 groups. So in summary, going through the summary of this last part. So if we're looking at primary vaccination, so the first 2 doses, the resting memory B cells correlate very well with antibody titers, and they are the source of the sustained long-lived memory response. They arise mainly from these CD71 activated precursors, and these are distinct from those that we see in chronically activated B cells, such as in HIV infection. And that booster vaccination and from booster vaccination, that the antibody and B cell responses are muted by recent infection, and that the interval between the 2 time points really is what correlates with the booster vaccine. So some of the things we're working on is we're trying to evaluate the effect of this repeated antigen exposure, whether it be through infection or vaccination, on the ability to respond, both at the B cell level and antibody. And we're also trying to better delineate trajectories of B cell responses to antigen after vaccination or infection, and hopefully that'll help us better understand correlates of protection. So most of this work that I presented today was conducted by 2 senior investigators in my group, Leda Cardava and Clarissa Beckner, with the help of many others, students, postdocs, and people who work in the clinic. My medically accountable senior investigators, Dr. Sneller and Ali Rai, who just joined us, a longtime collaborator, Taeok Chun. Of course, I was never be able to do this without the support from Dr. Fauci, who stepped down, unfortunately, a few months ago. We have a huge clinic who supports our work. A few other collaborators, both at NIAID and at the University of Maryland and the Yale School of Medicine. And of course, the study participants. Without their willingness to participate, we'd not be able to do this work. So thank you for your attention, and I'll happy to take some questions.
Okay, thank you, Suzanne. I think I will ask a question while other people may come to the microphone. So thank you very much for your presentation. For the last part, the second part, I think it's interesting to see how the distance between infection and immunization is important, and hopefully this will inform other vaccination strategies in the future. So, but this is not a question, it's just a commentary. For the first part, you mentioned the Tibet positive B cells that seem not to produce neutralizing antibodies. So what do we know about the transcriptional regulation of B cells that produce those broadly neutralizing antibodies, and can we use Tibet inhibitors to modulate the ratio between the good and the bad antibodies?
So I think a really interesting question, and not one that has, I think, a simple answer. But one of the things that we're thinking about is that it's possible that some people, when we look at just the phenotype of B cells of the few rare neutralizers that we do have. So basically when we talk about elite neutralizers, these are people who can make good potent neutralizing antibodies. Most of the time that doesn't help them because their virus has escaped that. However, why are they able? Like, is it just random? You know, I don't think it's random, but I do think that it's possible that they don't, it could be epigenetic, and that's, I think now there's some interesting data from mouse models that suggests that not all, and this is most T cell work, that not all exhausted T cells are like terminally exhausted, but they have a capacity to revert back. And it's possible that the same happens with B cells, is that you might go down this path, but you are able to reset. And I think that can be perhaps evaluated epigenetically, and we hope to be able to do that. We don't have many elite neutralizers on whom we have enough cells to do these analyses, but as the assays get easier and easier to perform on fewer and fewer cells, such as ATAC-seq and others, then I think that offers the opportunity to be able to answer those questions.
J'ai vraiment beaucoup aimé votre présentation. Merci beaucoup.
Merci.
I'm very interested in the second part of Petronella's question. What are the B cells making these non-neutralizing antibodies, which I happen to like a lot, particularly with the specificity of the cluster A region? So I know you're most interested in how can we make these neutralizing responses, but what about these non-neutralizing antibodies, these kind of junk antibodies being there that are maybe potentially problematic? Are you looking at that? Any specific signatures on those? Is this associated with, immune dysfunction to some point? Do you have any thought about it?
So I think those might come from people who have a kind of this underlying immune activation, and they come from more the short-lived plasmablasts, which may not have, that's been shown by others. There's a kind of like you're generating and regenerating, and so that's why maybe those binding antibodies don't wane as quickly as those that are, the more potent neutralizing antibodies. But to be honest, we haven't looked carefully into that aspect.
Because we do detect these antibodies in pretty much everyone we look at, even in people who are treated. And they are there. Sometimes our treatment brings them down a little bit, but without viral load, we still detect them. So do you have any idea what could be driving the presence of those?
Sorry.
Thanks.
Thanks, Susan. My question is regarding the energy that you observe for the B cell in COVID patient that has been previously infected. People were infected that with high hospitalization or low hospitalization, or what is the status of the patient?
Sorry. Pose ta question en français.
OK. La question est le statut de tes patients hospitalisés qui sont plus ou moins énergiques.
On parle du VIH ou on parle de...
COVID, seconde
COVID?
part, the last part.
OK, on n'a pas beaucoup de patients qui ont été hospitalisés. Ça, c'est des essais très tôt durant la pandémie. Alors, je n'ai pas présenté, je pense, de personnes qui étaient hospitalisées.
OK, so there is no statute about previous infection that is drastic or not drastic that may induce energy B cell?
Yeah, I think the problem is that you know, in the early days of the pandemic, we had access to people who were hospitalized at the clinical center, but they would get like 10 cc blood draw. So the B cell component was like a fraction of a fraction of a fraction, and we were able to get enough blood to do basically the baseline to start to develop the tools that would then allow us to do what I presented.
Yeah.
But one of the issues at NIH is that while we have a fantastic clinical center, it's not truly a hospital. It doesn't have an emergency ward. It doesn't. So there were still very few people hospitalized, you know, at NIH in a clinical center. So I think our experience is more limited, and it was more through these intervention studies that were done with different sites, of which NIH participated.
Yeah, because it's possible that during the acute phase there is a huge immunodeficiency, and it takes time to recover this immunodeficiency, and this is why during this period of like 6 months you observe that B cells are energetic, as compared probably to patients that has been higher to 6 months, in which they recover a normal immune system, in which now the B cell is capable to be stimulated again.
Right. I mean, I think that's very possible. Unfortunately, we didn't have the tools in the early days, and now we can't really go back because there's so few cells that have been frozen down to look, you know, do a deep dive into these different phenotypes.
In the context now in HIV field, not in COVID-19, in HIV field, do you think that maybe there is some compartment in tissue, for example, in the spleen, the situation of B cell differentiation or the activation of B cell is different to what we found in the blood, for example, about Tibet and... Yeah,
without a doubt. But it's just, again, you know, a little... We've had the luxury of doing lymph node biopsies, full lymph node biopsies. The fine needle aspirates can also be very helpful. Unfortunately, with the pandemic, that program was basically shut down, but we hope to be able to get back to it. But I think you know, where you can get enough B cells, 3 places, right? Bone marrow, full lymph node biopsy, and the spleen. We don't often have splenectomies that would afford us to do that.
Because it's like, because the dynamic of B cell probably is different to the peripheral blood. Thanks.
Thank you, Suzanne. So as a clinician, I will be interested to have your vision on, of course, time and from infection to vaccination, but for the immune imprinting, you know, the original seed, how do you think that may impact the effectiveness of subsequent vaccination with different variants?
Right. So if we take uninfected, so HIV uninfected, we do see evidence of diminishing returns, right, with each dose, and that's a bit concerning. So even though people respond, the same cohort, we're now seeing them through five doses. And each dose you get a lower boost, and it's also now being affected by, obviously most people have been infected, so it's a little bit hard now to really keep that cohort in line with good timing, and timing makes all the difference, I think.
But you may re-stimulate the initial
Yeah.
B-cell response and not the
Right,
latest
right,
one.
which is the other problem. But I think you also rise all boats a little bit. On the other hand, one of the interesting, although rather anecdotal, so one of our problems is that our HIV cohort, which is huge, was until recently not allowed to come in for their regular visits. So we're just now getting back to being able to do the intersection of HIV and SARS-CoV-2. And as I was saying in the previous session, our patients have most of them gotten their first 2 doses, but many have refused to get their boosters. But what we are finding is either their imprinting is different than an uninfected, or it's because they haven't gotten so many boosters and have been infected. We actually see a stronger BA4-5 response than in uninfected relative to the total. So it's a very interesting. We're just starting to delve into that. Is it that they don't imprint the same way? And that's possible. Or is it just their status, their vaccine status? And that I don't have an answer for today.